All right. We're about to start our panel discussion on drug discovery and AI. I'll introduce the moderator, and then we'll ask the panelists the first time you speak, take a minute and give us a little bit of your background. We appreciate that. It's a great panel. Our moderator is Laurent Audoly, and this is going to be an impressive one to try to do from memory because what a career. So PhD, Vanderbilt, chemistry, pharmacology, postdoctoral studies at UNC Chapel Hill, and an American Heart Association fellow, and then began a great path through multiple companies, Merck, Pfizer, in drug development, and then a path into serial entrepreneurship, both on the buy and sell side. Companies you may have heard of, founding CEO, Chimera. We have Cytovia, and now CEO and chairman of Parthenon. I hope I said it correctly. And during this career arc, we have 70 publications and patents. And then to me, the one that really strikes me, 21 different drugs, both small and large molecules, brought to the clinic, and of those, 7 medicines. So this is very impressive, and we're really honored. So please, Laurent. This is very important. I forgot. I knew I would forget something. Also, we have to talk about Northeastern, professor of the practice in both the College of Science and the Bouvet College of Health Science, and thank you very much for all your help with the AI Institute.
Great. Thanks, Jeff. Thanks so much. I'm super grateful for the opportunity to talk about something that is really a passion, and you'll see why, because it's an underpinning to how we can really break some additional potential here out of the biopharma sector. But I'm also grateful for the history, the perspective of the Barnett Institute, because we're going to talk about the future with the panelists, but we have to appreciate the present and also appreciate the past in order to build that better future. I'm also grateful that it gave me a really good excuse to bring together some super smart, accomplished executives in the sector, and also good people, and hopefully they'll think of me as their friends too, because I think of them as my friends. So without further ado, because they've got a lot more interesting things to say than I do, if I can ask all the panelists to come and join, please. You can welcome them all. Yeah, come on. Yeah, just if you guys, I'll go on the edge if you want. Yeah. Please, great. So before we get started with the content here, if each one of you, if you don't mind, please, brief intro and what connects you to the world of AI and ML, that would be awesome. Mike, please.
Hi, everybody. My name's Mike Crackower. I'm the CSO of a startup company in Boston called Ventus Therapeutics. I have a PhD in molecular biology and medical genetics from the University of Toronto. I did my postdoc training at the Princess Margaret Hospital and the Amgen Research Institute in Toronto as well. I have 20-plus year career in drug discovery, primarily focused on immunology, immunological diseases, fibrotic diseases, and now neurodegeneration. I'm here because I'm actually a biologist, okay? So I'm not a trained computational person. I'm not trained in AI, but I am very much a consumer of this space. And what I mean by that is our company, we are a small molecule drug discovery company, but we are powered by a physics-based computational platform that is machine learning enabled. And we believe that our approach can realize what is ultimately the vision of drug discovery in terms of small molecules, and that is to get to a drug product as quickly and efficiently as possible. And we believe our approach is ultimately has the chance to do that.
Great. Thanks for having us, Laurent. I'm Anna Marie Wagner. I wear a couple different hats at a biotech platform called Ginkgo Bioworks. I'm our head of AI and also lead corporate development, which basically means that my job is to make sure that any technologies that exist out in the world that our customers could benefit from, we find a way to work with them, whether that's through M&A, partnership, licensing, anything like that. I thought I was going to be an academic scientist. Very quickly learned I did not like doing lab work, and so switched to math. And one of the things that Ginkgo really does is trying to make people like me who didn't want to spend the next, dozens of years doing manual work, moving clear liquids around in a lab in order to advance the field, a platform to be able to really spend their time thinking about the science they want to be able to do, thinking about the way to run these experiments, and then using, whether you call it a cloud lab, a platform, automation, to be able to generate the data that we now have tools like the new advances in artificial intelligence to actually use in more interesting ways. So very privileged to be able to help there.
Hi, everyone. Enoch Huang. I'm at Pfizer. My background originally was in molecular biology. Became more of a computational biologist for same reasons. I didn't love the cold room and purifying proteins, but I loved doing the data analysis and molecular modeling. So I got into protein structure prediction at a time when it was mostly about bragging rights for academics. This is sort of like the CASP experiment, which some of you might have heard of. That was my training through grad school, which was at Stanford. Ironically, some 27 years later, now you have DeepMind and similar approaches winning the CASP competition. So now it's become very relevant for the pharmaceutical industry. I suffer from a little bit of imposter syndrome, so I don't describe myself as a machine learning expert, but one thing I am proud of is that I spotted the opportunity and the potential for machine learning far before many of our peer companies did. So my first position that I posted was in 2008 for machine learning research scientist for Pfizer. And back then, the competition for such jobs was non-existent, so I was able to pick from people who were formally trained in computer science and machine learning at the time, who came to me because no one else was posting jobs like this. And some of these folks are still at the company and leading larger computational science and machine learning applications groups. And maybe we'll get into some of what we do during the panel discussion. So look forward to it.
Yeah, so I'm Wengong. I'm a postdoc at Broad Institute. I got my PhD in computer science and machine learning from MIT. So during my PhD work, we're kind of among the first groups who are developing methods for drug discovery, AI methods for drug discovery. I developed this ChemProp tool package that are now widely used among many pharmaceutical companies to facilitate virtual screening of compounds. We're also through collaboration with Professor Jim Collins at Broad, we discovered a novel antibiotic that had broad spectrum activity using machine learning, and that was very exciting, kind of pulled me deeper into this field. Now at the Broad Institute, we are also thinking about how to develop generative AI methods for de novo antibody design and protein design, and we are I'm collaborating with immunologists at Broad to applying these AI-based antibody design methods for TCR-like nanobody design and cancer immunotherapy. Yeah, so really, it's my pleasure to be here, part of the panel.
Wow, so that's exciting. There's lots of firepower here, so I'm going to try to keep up here. But before we get going into some of the questions and perspectives here, just to set the stage a little bit, and I've got maybe 2 or 3 questions for the audience. I'll take the first one, it's the easy one. So median time from first patent to approval of a drug is 10 years, right? So we're in a super long cycle business. Here's a question for the audience. So what do you think is the approximate range of total R&D expenditures? It's in billions in 2023 across the entire sector. Just guesses, anyone?
180.
Got 100, got 110, 120, 130. 163 billion dollars. So that number is big number, obviously. But what's really interesting is that 10 years ago, it was half of that. So that starts to get interesting, right? So doubling of R&D expenditure in the sector, But the main thing that for me really drives these kinds of discussions and why I decided that it would be interesting to get involved a little bit more with AI is that our efficiency has basically flatlined over the past 3 decades, right? So simple math, right? It's a bit of an issue, right? You got 10 years, that 10 years is kind of a constant over time, doubling of the budget, flatlining of productivity. We need to do better, right? And so if you think about potential solutions, right? Better software, better hardware, reduce cycle times, handle bigger data, right? So that's how that door on AI ML potentially opens. And one last number here, and I had to write it down because it's actually remarkable, and I'm sure Anna Maria would also, and all the panelists would offer a perspective there. So in 2023, there was $13 billion worth of life sciences drug discovery deals with AI ML. $13 billion, right? In 22, that was half of that. It was a little bit less than half, 5.3 billion. So there's clearly a realization that there is a game changer there. We'll talk a little bit about what's real, what's overhype, what's around the corner, what's in the next 10 years. But we're talking big dollars, long cycle times, potential huge impacts for society, for patients and their families. So definitely this is a good topic here, and hopefully the Barnet will continue to play a role in that trajectory. So I'm going to start off with Mike, and I'm going to pull you in here on the small molecule discovery. We've been having discussions on what's real, what's not real, AI, ML, and small molecule discovery. And I remember our first discussion was like, thumbs down, right? It's like, there's nothing, it's all hype. But things have changed quickly. And so can you tell us what in your mind has changed? And also, you've contributed a great deal to that positive change. So what does that mean at Ventus in your experience? And then what's around the corner?
All right, great. So maybe I'll just start by saying ML is not new to drug discovery, as I'm sure many of you know. It's been around for at least 20 years. It wasn't called ML or AI, but we were doing it, and it was called things like QSAR, where we're using other types of statistical methods to try to pick the next molecule to make as you're trying to optimize something with drug-like properties. We've also all heard about systems biology. Strangely enough, nobody seems to talk about systems biology anymore, but we're still trying to do that. Now we're calling it machine learning, AI, and trying to use advanced computing power that we have now to do it better. Let's call it that. So it's not new. What we've seen in terms of things that are new and breakthrough are things like that was just mentioned, AlphaFold. This is an absolute breakthrough. It shocked a lot of people that you could do this. It was actually, if you fundamentally think about it, not that difficult of a problem if you just figure out how to train the data set, right? And the recognition that you could use natural language models to do this. And it's like, oh, this is actually pretty straightforward. And so this is definitely real. But is it having impact? Not yet. And the reason why is because it's not high enough resolution to actually have impact in drug discovery. We have to understand side chains and rotamers and protonation states to do what we do. Having the backbone is a good step, but it's a step towards where you ultimately want to go. You can use the same approaches to help train other aspects of drug discovery, not to deviate too much from small molecules, but this is essentially what people are using to try to design antibodies using ML. And some of these are working. Are we doing de novo antibody design? No, we're not. Will we get there? Maybe. It might be a tractable problem. The way I see this is there is a lot of application for machine learning. It's incremental. It will be incremental. I don't believe we're going to see something that just completely changes the way we do things in a dramatic way. I don't believe it will ever be done without wet labs. Sorry for all you people who don't like wet lab work. I love wet labs. I never had as much fun working in a wet lab. I think that's absolutely always going to be part of the process. And I think some of the advancements that we're seeing now are actually wet lab advancements in terms of chemoproteomics and what we can do there to really identify new targets and new binding pockets and things like that. But there's a lot of hype. And that's one of my concerns, is that there are things that people are claiming they can do in drug discovery with machine learning that they can't. And they can't because it's not possible. And I worry about those kinds of things because that excitement will come tumbling down, and it will have negative ramifications on the industry. Saying a little bit about we do, so our approach is very physics-based. At the end of the day, how a small molecule binds to a protein, that's physics and math. And why we're more interested in the physics base is because we believe this is a universal. Physics is universal. It's not the physics of how a ligand interacts with the protein is going to be the same regardless of the protein and the ligand. Therefore, it's something you can actually get a big enough training set to train on this. And we think that's the future of how you get faster and better making small molecules. And so that's the approach we take. We're very driven by a solvation-based physics, so water-based physics, which is the fundamental driver of how proteins interact. It's this, and why they interact and how tightly. It's basically displacement of water. And so if you can understand that well enough, you can actually get enough data points to train on it. And so that's our approach. What we do right now is we do things that can help our process go quicker. So things that do work already, but are labor intensive, and you could train to make it more automated and so on. So that's where we're applying things right now.
Thank you very much, Mike. Enoch, thoughts on the small molecule front?
Yeah, I think I agree with everything Mike just said. As far as the roots of what we call modern machine learning started with statistical learning and learning from data like the QSAR models. And so that's where the success that I agree with you, Mike, it's been around for decades. And when I said I started hiring people in 2008, it's precisely because we had a wealth of institutional data that was ripe for exploitation using statistical learning, now machine learning models. And these are the properties that all small molecules need if they have a chance to be an orally bioavailable drug. Properties like permeability, metabolic stability, solubility, and we have some 18 to 20 assays which are routinely run to characterize all molecules with its potential. And the beauty of this data set is that it covers a wide range of chemical space, so it generalizes well to compounds that you haven't yet made. And so this authoritative data set has become sort of the underpinning for classical machine learning methods like ensemble tree-based methods. We could go to more advanced ones, but generally these ensemble tree-based methods perform very well for the purpose at hand, which is to weed out less productive design ideas in virtual space, things that you haven't yet made but could make. And so this allows us to have fewer, more productive rounds of design because you're able to sample a more productive area for bioavailability in the end. And so there are like 4 ingredients which any machine learning endeavor in biopharma needs to generalize. You need to have proper training data that's curated and processed, prepared, if you like, for machine learning input models, input to machine learning models. You need to have ways to deploy these models that's usable by scientists, in this case, medicinal chemists. You need to have enough compute power, and you need to have software so that it can interact with the lab scientists, like I said. And so those, you can extend it to biologics or genomics, but these are the key ingredients for this area. So we're really proud now that, the system that we've designed over the last 20 years, it's used 6 million times a day to do calculations, which is over 2 billion calculations per year on compounds that we could make, and then we're triaging those ideas in a computer. These assays, these computational assays, are 75% of the time within 2-fold of the experimental value, which is fit for purpose. You're just looking at potential designs. And so this is a well-oiled machine. There are other applications like biologics that we're just getting started. We're very proud of the work we've done here. Sometimes it requires generation of training data specific for the task at hand, which we kind of got for free when we started machine learning work in small molecules. But we have to be intentional, not just about the data set preparation and generation, but also the techniques. Now, this is where the rise of deep neural nets, like convolutional neural nets, and we published some of this more recently, where traditional statistical-based methods, where you predefine the features as input into a tree-based model, no longer do the job. You have to do end-to-end learning where using techniques that were invented in computer vision, can be applied towards biologic discovery, learning what patterns in the data that you can only discover as part of the training process as you learn the weights and the biases on the algorithm. And then the output then become fit for the purpose at hand. Properties like viscosity or polyreactivity or stability. So these are the properties in large molecules. Now we're starting to get a good grasp on, just like we did for small molecules 2 decades ago.
Thanks, Enoch. And I would also invite the audience to take a look at the Panelists' PubMed. It's super impressive, and there's a lot of threads here that if you're interested, you can pull on and take a look at what's on PubMed with these authors. So maybe taking a step back here, and Anna-Marie, just getting your perspective on data and setting up the right framework and the right strategy around how do we best put in place a data strategy that's going to be helpful. So we've heard from Enoch about data. Can you please comment on what you think is missing, what works well in the world of data strategy?
Yeah, absolutely. I feel like I spend about 80% of my time preaching the value of high-quality data these days, and completely agree with Enoch in terms of the way that we think about the ingredients, the universal ingredients for AI. What's happening now in biology is really interesting, where a lot of folks from the traditional AI ML space and kind of language, human language, are moving into biology because they recognize the similarities, right? It is a language, it's sequential, you can use a lot of the same techniques, and it is valuable. And they look at the data sets that exist, and the first thing they say is, oh my gosh, there's so much data. There are 1 trillion tokens already in these databases that I can access. But there's a lack of appreciation, I think, sometimes in that transition for the sheer complexity of biology and the relevance of that data to the challenges we're actually trying to solve. The reality is that the data that exists both publicly and internally at companies is quite sparse. It's highly clustered around the types of questions that folks were generating that data to answer. The entire R&D process is designed to minimize the amount of experimentation we need to do because it's so expensive. And so what you end up having is this really steep funnel where you collect shallow amounts of data about lots of things, but then you're only collecting deep, high-quality data about the things that work, because it's just too expensive at most places to generate that data about your broader search space. So what ends up happening is we only have data on the positives. We're missing the 99.999% of data that didn't work, and you need that data to train a model well. There's been a lot of research in the traditional ML community around the right balance between these ingredients, specifically between data and compute. What a lot of folks are doing is trying to overcome the data limitations by just applying more horsepower, more compute. And that does create benefits. And if you look at the AlphaFold 3 paper that just came out this week, what's interesting and actually kind of exciting about that is that they got some pretty interesting advances in that paper. They expanded AlphaFold from just being proteins to all biomolecules. They started showing a lot of functional prediction at, much, much better than the existing kind of state of the art. That's exciting. But the only thing they really did is they applied more compute and they applied different types of algorithms. It's all still public data. And so I am both excited, I'm excited about what is already happening, but also recognize that we are so far off the optimal frontier of balancing data and compute that the ROI for every incremental high-quality data point is much, much, much higher than the ROI from every incremental, kind of equivalent unit of compute. And so if we can create tools that allow the community broadly to access higher quality data that has been generated for the purpose of training models, in other words, it's well distributed, it is multimodal, it is high quality, it is consistent across batches, the returns that we can get on those types of investments are enormous because the tooling is there. The tooling is sort of caught up, and what's missing on a relative basis is the data.
That's great insights. Maybe we'll come back to that, and maybe with an eye towards how can a research institution like Northeastern join forces with the industry in order to create some win-wins here.
I have ideas.
Awesome. That's great. So, Wengong, you've published both on small molecules, on large molecules, specifically antibodies or antibody-like molecules. Can you provide your perspective on some of the key differences on the, let's put it this way, the impact that machine learning-based approaches will have on drug discovery with these different modalities. So can you compare and contrast small molecule to antibodies using ML to discover new structures, new molecules?
Yeah, that's a great question. I should say actually there are a lot of similarities between small molecule drug discovery and antibody drug discovery from a machine learning standpoint. But there are definitely a lot of differences from manufacturing and from assay development. If you look at AlphaFold, now the method is able to do protein ligand, structure prediction, antibody antigen docking, and protein-protein or protein nucleic acid interaction. So it seems like this emergence of foundation models that are not only just in terms of the compute or the number of parameters, but also like in terms of the breadth of the interaction, like biology that it could capture, is starting to get interesting. That imagine in the future that you could think about kind of using AlphaFold 3 or equivalent to inverse design small molecule binders or antibody binders. I feel there might be actually a unified theme, a unified framework for this, and of course that will take a lot of effort from the machine learning community to really make it happen. Also collaboration with industry and with experimentalists. I feel there's actually a possibility for building this unified framework. So I wouldn't say there's like real key difference on the methodology, but there will be a key difference in terms of how this closed loop pipeline will look like. For example, small molecules are relatively hard to synthesize, right? So you need to, at MIT we have built this automated synthesis lab for small molecules that actually took us like several years to make it happen. Whereas for antibodies, you have yeast display or ribosome display libraries that could rapidly screen for billions of antibodies and you can relatively easy to manufacture them as long as you have these are relatively stable and have less immunogenicity, right? So I feel the set of criteria to look at will be different, right? For small molecules, you have to look at toxicity and other criteria, whereas for antibodies, you have to look at immunogenicity and developability, right? So the set of criteria will be different, but the methodology, like from a machine learning standpoint, will be actually relatively similar, in my opinion.
Thanks, Wengong. So maybe pulling on one of the comment that Mike made on de novo antibody discovery, which is kind of a big deal, right? When you think about it, right now it takes about 2 years using, let's call it old-fashioned approaches to go from idea to a pretty robust antibody lead. So if we can use a computer to do that in a week, or a day, boy, that takes out a lot of headaches, right, from the process. So I've sat in rooms and panels where there's always, that one or two individual that says, oh, my company does de novo antibody discovery. And I go, can you please explain me what you do? And it never makes sense. But maybe Wengong, you've got your hands on the pulse here. Can you offer a perspective on that? And if we are at that stage, and if we're not, what would it take? And then, Mike, maybe also, can you sort of do a ping pong with Wengong on what would it take in your mind also to get us to the place of de novo antibody lead discovery?
Yeah, sounds good. I actually talked to quite a few antibody design companies like Epsi and there's Nabla in Boston. They are doing very exciting research, I would say, and I'm quite fascinated about their closed loop facilities that connects machine learning and rapid testing. So I can comment, there are several levels of, let's drop the word de novo, but several levels of antibody design pipelines. You could generate a ton of data for the target, for the antigen of interest, like in the most most common target that people have been playing around is HER2, which is related to cancer a lot. And there's a lot of East display library data that has been generated. And you can train machine learning models to distinguish what binds better than the others, like what antibody sequence has the best binding affinity. These, I would say, kind of it's like level 1 level, antibody design campaign where you generate data, you train a machine learning model, and you try to tweak binding affinity to the max, right? And this is very valuable, I would say. I wouldn't say that this is, we should continue to strengthen this effort, but I wouldn't call it de novo antibody design because you are generating data and your method will work for that particular target, right? So I can imagine that the ultimate sort of level would be you have a platform that you could design for many type of targets, not just for one target, and you don't even need to generate the data that binds to that through experimental approaches. You just by looking at the data that you have generated in the past, or like the crystal structures in protein data bank, and use it to design antibodies that binds to your new target that you have never really did any assay. That would, I would say, is like de novo antibody design, where we haven't really get there yet. But if you look at the advancement of AlphaFold 3 and its capability of modeling, like its ability to model antibody-antigen interaction actually improved by 30%. That's quite a bit of improvement. And perhaps there will be an avalanche in maybe we are soon at a point of breakthrough that these powerful structure prediction models could enable de novo design of antibodies. But I think the current existing work that has been published so far either has to rely on the data generation, or you can design a binder but with relatively mediocre binding affinity. So which means you need to take several rounds of affinity maturation that involves iterative sort of experimental validation and improvements. So I just want to also mention that I think, physics, even though a lot of the existing methods like AlphaFold kind of try to leverage data and these crystal structure data, or like evolution, like multi-sequence alignment to predict structures. But I believe that there are ways, actually I'm personally interested in exploring ways to integrate physics into these machine learning models to kind of use it as a way to pre-train or these methods to generate good representation. So I think this is an open area of research that I think will also increase, and because the need of generalizing to new targets that we have not seen. Yeah. So just wonder if you have any comments on that too.
So I agree with a lot of what you're saying. I think so. I actually work with some companies that are trying to do machine learning-based antibody design. What they can do effectively right now is make new sequences for known folds, right? That very straightforward how you might train on that, very similar to an AlphaFold. They cannot design new antibodies yet that have completely new folds. But you can imagine that's trainable, right? If you understand protein-protein interaction and you can train enough of that, you could train on this. So I think that is an area where the problem is tractable. We've talked about libraries that have billions of sequences of antibodies. That's very tractable. It's very different than small molecule, where you're talking about 10 to the 63 potential small molecules, which makes that impossible to actually train. So you have to use physics-based approaches. But I just like one thing that I think is really important, though, and when I started to work with these companies, and they said, what should we do? Like, what are the problems with antibody drug design that we could address? And honestly, my first answer was like, I don't know. Because you can very effectively screen for antibodies. There are great platforms that can do this. They can find very rare antibodies. You can do high throughput sequencing, find those very rare epitopes, and that works really well. So one of the challenges is actually, and I think for a lot of ML approaches, is what are we trying to solve? What is the actual problem we're trying to solve, as opposed to I can do the same thing, incrementally better or maybe just different, but I didn't solve a problem. And I think that's one of the questions that's going to come in antibody drug discovery, because just getting a binder to a protein, we can do that. We can do that very well. So can we think about applying ML to getting bifunctional antibodies, to getting trifunctional antibodies, to getting conditional antibodies? This is a whole order of magnitude more complex kind of proposition, but this is where it's very difficult to do in a wet lab, and you can imagine ML being able to have real impact in this space.
That's great. Thanks a bunch. Enoch, just really briefly, because then I want to come back to Anna Marie. So what can we do today to actually improve the drug-like properties of antibodies so that we're not using just lab-based approaches?
Right. I think what problem we're trying to solve, the question that Mike posed, is the critical one for antibody discovery, because from screening you can get the affinity relatively straightforward. So the proposition we were challenged with is viscosity, because it requires a you need to generate a lot of purified protein in order to do that assay. So often that's the rate limiting part of the discovery cycle. So if you had a computational surrogate that allowed you to predict viscosity, maybe not perfectly, but good enough to build that into that design, then when one round of design, you can have something that is just not only a high affinity, but also has excellent viscosity. So we were able to take one round of design entirely out, and therefore accelerating the process of getting to the clinic. So I think you have to look property by property in antibodies, but generally not affinity.
It's great insights. Anna Marie, can we come back then to the data problem, the culture, the frameworks that need to be put in place, maybe with an eye towards private-public partnerships?
Absolutely. So one of the things I've observed, and maybe just to give you a little bit more background on Ginkgo, most of our customers, most of our revenue comes from large pharmaceutical companies, large international conglomerates, places like we've got a big collaboration with Pfizer, Novo Nordisk, Bayer on the crop science side. And they view us as a way to dramatically scale up basically their data generation capabilities in service of discovering a drug or discovering a new crop ingredient. The issue is that what ends up happening is it's kind of reinforcing an existing mismatch or just gap on the infrastructure that is available to the open science community. These very large companies have really strong internal R&D teams spending hundreds of millions, if not billions of dollars on this type of research. A place like Ginkgo has poured well over $1 billion building infrastructure that these companies can now use to supercharge that. What we haven't seen is that type of infrastructure being made widely available to basic research. It's being deployed to really important problems. Like, we're really proud of the work that we enable for our customers, but one of the things I've been spending a lot of time on recently is how we can bridge that gap. One of the issues is funding. Like, generating this kind of data, even on a really efficient platform like Ginkgo's, and generating enough of it that it's really useful, is expensive. It's a physical process. There's just, it's going to take time before that comes down far enough. And if you look at the way funding is distributed to basic science, it is distributed in these little tiny pockets of, $1 million grants here and there. And so, and there are folks that are, trying to fix that. You've got the Chan Zuckerbergs and the ARC Institutes and the Allen Institutes and some of these larger foundations that are trying to pull the funding together. But even then, when the funding is pulled together, it's still distributed into these smaller projects and smaller grants. And so one of the things I would really love to see is a large, concentrated effort to generate data that can be a shared resource for the community. Because one of the issues you see, even with the approach right now is to solve it through collaboration. Like, let's put out a call for lots and lots and lots of labs to generate data, and we'll pool it together and we'll try to clean it up. That is a very, very hard thing to do. It is admirable. We want to support it, but the batch effects are huge. The metadata is very, very, very hard to capture appropriately, and so you end up spending as much time and money and energy trying to get that data to reconcile with across labs as you do trying to get any benefit out of it or collecting it in the first place. And so I do think there's an opportunity to invest, like almost a human genome project scale endeavor, as as industry, as academia, as government, to create a very high quality data set. Again, I think the money is there. I think the infrastructure exists, and it's just about bringing the ambition together and the ideas together on, like, what do we want? Like, what would really change the course of all the science that your students are doing, so that they're not spending their time moving clear liquids around a lab with their PhDs, but they're spending their time thinking about what data would be useful and then advancing the field with that data.
Fantastic. So do we have a minute or two maybe for questions here, if there are any?
We do.
All right.
Can we hear me? Okay, perfect. I'm going to take advantage of having the microphone and I'll try and make this quick. In 1992, I thought the coolest thing would be to see how a molecule would stick to a protein. So then I took molecular mechanics, and then I took quantum mechanics, and then I taught quantum mechanics. And then I went to every single training that Schrodinger offered. And then a day finally came about 3 years ago when the predictions, physics-based, were within a kcal per mole in the right system, and they will not provide the license. And I organized the site license here. They would not provide academics with the site license. So basically, I wasted 32 years, unless you guys have what do we do now? What are, docking, it's to your point, Anne-Marie, that it's such an important resource. Companies are being spun out, and we don't have it.
You're talking about FEP.
Yeah,
Plus.
exactly.
Use GrowMax.
Pardon me?
GrowMax.
Open source.
Perfect, thank you. Other questions? We have one more.
That's great.
So it's really great to hear, all your insights on AI. I guess as I'm listening to it, and you have these computational tools that can design these molecules, the challenge to me, or at least the question I have, is when do you get intellectual property involved? Is it at the AI stage, or do you have to wait until it manifests as a protein or a molecule?
I mean, so we have a lot of computational capability. We have a lot of algorithms that we use that are proprietary, but we don't try to get copyrights on those because at the end of the day, someone's going to be able to get around that. And so we really keep these as trade secrets. And so for us, the IP is at the small molecule stage when typically, no different than any other company or pharma, when you've got enough data that you feel like you're not going to prior art yourself. This is one of the key things where I file a patent and I didn't have that many molecules in it, but I actually want to patent the next one, but I can't because I've prior-arted myself. So you have to be careful about the timing of that and how you split apart your various subseries and things like that. But yeah.
I have a perhaps controversial take on this, or unpopular. I think the basis of competition in pharma is going to have to change. It's going to take time. There's going to be a lot of resistance to it, and I'd be curious for your thoughts on this, but it's going to have to change. Like probably the biggest use for AI right now in the industry is working around other people's patents. And as AI gets better, it is like, and again, this is a good thing for the world. Like it will be easier and easier and easier to develop things that work. We'll still be bottlenecked by going to clinic and doing all of that work. It'll still be hard to distribute and price and do all the things that make these businesses. But the promise of AI is that it does get easier for people to design things that work, and that will make it harder to protect these molecules and these designs. And so it'll be really interesting to see what changes, like what breaks. And again, you could see patent law getting stronger there and covering broader spaces. You could see it just, again, like the basis of competition could also change. And I don't know what that looks like, but I think it's a really insightful question, and I don't know that anyone really has a sense yet of how that's going to shake out.
I mean, companies like Pfizer, Merck, GSK, I believe we are companies to discover and deliver medicines for patients, and that is where the intellectual property is, to Mike's point. I see, personally, everything that is before phase 3 is being pre-competitive, and all the tools and approaches and technologies and methods associated with that. So it's the speed and success of phase 3 that the basis of our competition of these companies. And so similar to what Mike said, I think freedom to operate, access to these tools is paramount for us. So for us, if we develop a new computational method, and we do occasionally, we publish it. We deliberately publish it so we will have continued access to use tools like this because no one else can patent it because of the prior art that Mike referred to. For us, that's not the basis of our competition.
Yeah, actually, for us, we also, my research tools are always, published and open source, and actually it's adopted by a lot of pharma companies to use. And I think that's because I'm from the academic world, so I always want to, push science through open source, and the IPs are more or less on the actual discoveries, and for the antibiotics that we discovered before, we do have patents to protect them, but the technology is actually just released and allow everyone to use, and actually it's being widely used.
Protein molecules are highly dynamic, and those dynamics are absolutely fundamental, essential to their biological functions. And so far, artificial intelligence has been successful only at predicting static structures. I would love to hear your thoughts about extending artificial intelligence predictions to protein dynamics, particularly in relationship to molecular dynamic simulations as done by Dee Shaw's research group and others. Can we use those simulations to train models? Can we do this better?
So I think that's a great question that I don't know the answer to, so we use extensive molecular dynamics. We're one of the only companies that do fully unrestrained molecular dynamics. We certainly use machine learning to do protein prep. That we have that as fully automated, which you need to do. And I think what you're saying in theory could make sense, but I think the complexity of that problem is going to, it's a data problem that I think is immense in order to get enough to really train on dynamics. But I couldn't agree more. If you're not using molecular dynamics and doing structure-based drug design, you might as well not be bothering because you're working in an artificial system.
Actually, there are a lot of recent work on modeling, building machine learning methods to model protein conformational changes. There is a paper just published in Nature earlier this year using, changing the multi-sequence alignment in AlphaFold and kind of play with it to generate, diverse set of conformations for a given protein. Wouldn't call it protein dynamics, but models, the conformational changes or ensembles of the proteins. And there has been also a big decent data set generated for molecular dynamic simulation of protein ligand binding. That's around 15,000 trajectories of different protein ligand systems with each 100 nanoseconds and 100 steps. So these are data that has been kind of generated to train machine learning models to learn molecular dynamics, to accelerate molecular dynamics, to look at how the protein changes upon ligand binding, are there any cryptic pockets, and understanding the protein function. So there has been a lot of interesting progress there. I would think would recommend you to check out. Yeah.
And I'm not sure how technical this audience is, but just to make sure that everyone understands the importance of this question, one of my favorite biology is magic kind of stories is in all of our cells there's this protein. It's called ATP, ATP synthase, and it is what powers your cells. It is inside a 10 nanometer diameter, that's the diameter of the protein, is a rotary motor that spins at 10,000 revolutions per minute, which is faster than a 747 jet engine. And that is what allows all the electrons to move around and actually power your cells. It is magic, and it is programmed in DNA. And we are nowhere close to actually understanding that stuff by looking at the code. But it is freaking magic if we can figure this stuff out.
I just had one comment to that, just to extend upon that. The key too is, if you're not doing it in the cellular context, which none of us are talking about, we're talking about this at an individual, isolated protein. Most of what we're doing is not real. It's real at a very microscopic level, but at a protein level, the proteins are moving immensely when they're interacting with other proteins. And there's many, many examples where you can get pockets in a cell that don't exist in an isolated protein. And this is what we actually have to solve for, putting things in a much more, adding these protein-based approaches, molecular-based approaches, but in the context of a cell.
A little less scientific question. After you discover all these drugs, what are some of from your experience, what are your visions of the changes in clinical trial processes that how will AI influence the clinical trials stage?
Great question, and in 1 minute or less.
Yeah, so I'm going to, and I think this is a great tee up for the next panel on clinical development, and we didn't address it today at all. So we're going to try to do this in 30 seconds. One of you can jump in.
Fingers are pointing at me. So I'm not a clinician and I'm not involved in clinical trials, but artificial intelligence at my company for clinical trials is actually taking a lot of the manual and very repetitive processes that require a lot of human effort and streamlining it using computer techniques such as large language models like ChatGPT. Are there ways to use that technology to do medical writing or to prepare regulatory documents quicker and with less manual steps than before?
I think that's absolutely right. That's one of the key opportunities. In terms of making trials better, people are using machine learning to monitor their sites, for example, and seeing if something's unusual at one of the sites where the trial's going on so that they don't find out after the fact that there was a big problem, but they find out in real time and they can fix those problems, because those can be detrimental to a clinical trial, right? The clinical trials are, I'm sure everybody knows here, are insanely expensive, and you get one shot. It's not an experiment, right? You don't get to repeat it and say, oh, I'll do it a bit different next time. If you don't get it right the first time, it's all done. Yeah.
Great question. Thank you, everyone.
Let's take a minute and thank a great panel.